Dr.YSRHU, an eminent entomologist who has vast experience in coconut crop, and she has worked on the management of coconut pest for 25 years at Horticultural Research Station, Ambajipet, which is the only one research station working on coconut. And Madam has identified a new pest in mango also, that is Leptopteran mango fruit borer, causing havoc in mango belt of Andhra Pradesh. For the first time, she has identified that, and she has investigated on the bioecology, larval behavior, nature and symptoms of damage, feeding potential of a mango fruit borer. And she was also deputed by Government of India as an ITEC expert to the Government of Trinidad and Tobago, West Indies, for 1 year to do research on red palm mite, which is a serious pest on coconut there. And she has estimated red palm mite damage and occurrence of natural enemies of red palm mite, and established protocols for mass rearing of red palm mite on coconut and its predatory also in the laboratory. And she has trained several staff in the department on lab techniques. Later, she has worked as associate dean at College of Horticulture, Venkatramanagudam, and successfully facilitated and involved in various academic activities. Presently, she is working as Dean of Student Affairs in Dr.YSR Horticultural University, Venkatramanagudam since 2016. And Madam has received several meritorious awards for her outstanding contribution in the field of entomology. Almost 10 awards she has received. A few of them I can mention, like Dr. YSRHU Women Scientist Award in 1996, Meritorious Research Scientist Award in 1999, Azra Fellowship Award in 2000, Best NATP Research Project Team Award in 2004, Dr. B.C. Dev Memorial Award in 2005, and AP Scientist Award in 2010, and Padma Shri Dr. I.V. Subbarao Puraskaram in 2016, Anand Prakash Award in 2018. And dear participants, I am happy to share with you all that one more feather is going to be added to her crown of awards. Madam is going to receive Lifetime Achievement Award from Dr. Vasanthras David Foundation in the month of December 2020. And she has carried out 12 externally funded projects to a tune of 272 lakhs and developed and strengthened infrastructural facilities in entomology laboratory of the university. Madam has published almost 86 original research papers, 2 textbooks, 3 chapters in textbooks, book chapters, 71 popular articles, 14 technical bulletins, 19 brochures and pamphlets. And she has participated in 38 symposia conferences, symposia as well as conferences, and AICRP on palms workshops, presented papers, and also attended one summer institute program and 5 other training programs also on the management of coconut. And Madam has developed new technologies in coconut. We can mention like mass multiplication technique for the parasitoid of coconut black-headed caterpillar, and it introduced it in the new ecosystem, that is east coast of Andhra Pradesh, from and also with persistent recovery up to 30%, and standardized breeding techniques of different bioagents of coconut black-headed caterpillar, standardized the screening technique of rhinoceros beetle for identification of baculovirus infection, developed integrated pest management technologies for the management of important pests of coconut, that is coconut black-headed caterpillar, rhinoceros beetle, red palm weevil, etc. And she was also involved in release of new varieties and hybrids. We can name them. Few of them are Godavari Ganga, Double Century, Gautami Ganga, Kira Bastar, and also Kalpa Pratibha. And Madam is also a life member in many scientific societies, and she is vice president for the societies such as Applied Zoological Research Association, Association for Advancement of Pest Management in Horticultural Ecosystems, and Society for Promotion of Horticulture also. So this is a brief introduction of Dr.A.Sujatha, Madam. Madam, on behalf of the organizing committee and also all the participants, we heartily congratulate you for going to receive this Lifetime Achievement Award in the field of entomology. Congratulations, Madam. So I invite Madam to deliver a lecture on the management coconut pests as well as their management. So over to Dr.A.Sujatha, Madam.
Thank you, Dr. Sujatha, for giving a good opinion about me to the audience. And I feel, first of all, I would like to extend very good evening to all the dignitaries who are participating in this platform and the big group of student community. I'm very happy to share my experiences in coconut pest management with the students on this platform. Hence, I acknowledge the efforts put by the faculty of College of Horticulture, Venkatramanagudam, and the organizing committee headed by Dr. Uma Jyoti, Associate Dean, and all the team members who managed to conceptualize this coconut graduate program, under which I have given a time to project my experience on coconut pests. Thank you once again. I would like to start our presentation. I will go with my own screen sharing. We know that coconut is an important plantation crop in India, which is being grown in 90 countries and territories, and leading by Philippines, Indonesia, and India is occupying third position in area, with the area that is 1.9758 million hectares with a production of 21665 million nuts. But even though it is in third position in area and production, when you see the annual nut production, India stands first in the world with 13 billion nuts. Within India, if we see the scenario of coconut growing area, Kerala stands first, followed by Karnataka, Tamil Nadu, whereas Andhra Pradesh, with its area of 1.1 lakh hectares, with 1377.53 million nuts, stands its fourth position. But when it comes into the productivity per hectare yields, it stands first in per hectare yields productivity. Within the AP, East Godavari shares more than 50% of area, that is 5741 hectares, followed by West Godavari. The figures are being changed year by year. Latest, Srikakulam is taking its second place with 25,000 hectares, followed by West Godavari. When we see the production scenario of coconut, there are many constraints for good productivity of any crop, likewise coconut. Coconut being with a unique name, Kalpavruksha, it is perennial in nature with lots of foliage, abundant foliage, which assures uninterrupted food supply to the insects, besides its perennial nature. So that is the main reason coconut crop suffers with a lot of insect fauna and non-insect pests also. That is more than 750 insects and non-insect pests are recorded as per the literature. But among them, we can name few which are major constraints for coconut production within India, as well as more particular to our Andhra Pradesh state. These pests will attack any part of the coconut tree. That is, there are foliage pests, there are trunk feeders, root feeders, nut attacking insect pests, and other non-insect pests. Likewise, they can attack any part of the coconut palm and causing the reasonable damage, ultimately resulting in yield losses. So we will go one by one in this class. Number one pest. It can be coconut black-headed caterpillar, that is Opicina arnosella, rhinoceros beetle, Oryctus rhinoceros, red palm weevil, Rhynchophorus ferruginous, slug caterpillar, Macroplectra narraria, Eriophyid mite, Asteria gurroronis. The list goes like this. Then white grub. It is not much in our Andhra Pradesh state, but it is more prevalent in Kerala, Karnataka, in those states. That is Leucophilus coniophora. And nowadays we are seeing Rugose spiraling whitefly with large outbreaks in all in South Indian states. That is Alirodicus rugioperculatus. And then other important pest, but it is not present in India presently. But earlier it was there. Red palm mite, that is Rauviella indica, which attacks coconut foliage. The other one, coconut leaf beetle, this is also not present in India, that is Brantispa longissima. But they are waiting to enter to India, so we must be very, very cautious to know the details about these pests. So when we look into the coconut black-headed caterpillar, this pest is more prevalent in coastal areas and backwater tracts, and especially in the coconuts which are being grown on fish pond bunds. These are more suitable ecosystem. That is near water courses, the coconut plantations suffer a lot with this pest. So these will attack all ages of coconut palms, and they are having alternate hosts also: palmyra, talipot palm, wild date, and ornamental palm. All palms group will suffer with this pest. And then a specific character of this pest is the infestation starts from the lower whorls of the leaves. And the other characteristic feature is the larvae feed on the chlorophyll content of the leaf laminar portion. It will scrape the laminar portion, leaving only midrib of, epidermal layer of the leaf. Because of this, the leaf will develop dried up patches, and when all these dried up patches coalesce, we can see the burnt up appearance of the palm. So because of this foliage drying, so all the leaves will droop and bunches will buckle and immature nuts will fall down, leading to the yield losses finally. If it is occurring on the nursery in the seedling, so the growth will be retarded, and sometimes we need to go for replanting. When you go in detail, life cycle of this coconut black-headed caterpillar, the eggs will be laid in the leaf itself, and they are very round, and they will hatch within 5 days. Before hatching, they will turn into pinkish color. So the larva will be with black head. That's why it is called coconut black-headed caterpillar. And the pupation will take place within the fold of the leaflet, and then adult will emerge within 12 days after pupation. So total life cycle, it will take nearly 66 days on an average. So the management of this pest, we can go step by step. In severe case of infestation, when all the foliage was dried, the infested and dried leaves can be removed and burnt for eliminating the pest stages in the foliage. And then when the pest is at lower level, we can release parasitoids according to the stage. That is, we have two larval parasites. In the early stage, BRCA inhibitor. In the later stage, Goniosus nepenthes. In pupal stage, we have Brachymeria nosati. So these parasites can be released to suppress the pest population when it is in the lower level. So the other important thing is we need to conserve the natural enemies of the black-headed caterpillar. Those are lot of spider fauna is existing in our coconut gardens. So the other important predator is Perina nigrolineata, which is a carabid beetle which feeds on the life stages of the coconut black-headed caterpillar. Then when the moth stage is prevailing in the garden, if possible, if garden height is less, we can go for spraying with dichlorovas at 0.02%. And then in severe cases, if the larval stage is present in the foliage, we can go for root feeding, which is very successful method with monocrotophos 10 ml plus 10 ml of water. We need to trace out at the base like this. And a pencil size dark red brown root need to be selected. And with this, with sharp knife, we have to cut lower end of the root. And then empty polythene cover is need to be inserted. And then the liquid has to be 10 ml monocrotophos and 10 ml water is placed in this cover. And this has to be made like this. That arrangement in such a way, the last drop of the liquid should reach the tip of the root. This arrangement will take 24 hours on hot sunny day to absorb total liquid. And the pest will be controlled after 7 to 10 days. Here, the important caution we need to be followed is immediately after root feeding, up to 45 days safety period need to be observed. If any tender nuts should not be harvested before 45 days. So with this IPM, we can manage the coconut black-headed caterpillar. The other important pest is the rhinoceros beetle. That is Arctos rhinoceros. This will attack the palms which are having less than 10 years age. That is, they are more prone to this attack. They will bore into the spindle portion, that is unopened spindle portion. The beetle will bore and cause by eating inside the tender tissue. So this oblong shaped holes it will cause. And when it feeds on the fibrous material inside the unopened bud, the chewed up fibrous material will come out from the hole like this. And then if it is repeatedly attacks this portion, then mechanically the tender portion of the palm will be damaged. Sometimes death occurs. From this tender leaf, when it unfurls, when it is matured, and if we see this geometrical cut, this is the typical characteristic symptom of the rhinoceros beetle attack at its early stage. When we see the life cycle details of this one, the eggs will be laid in the decomposing compost pits or sometimes the bole region of the coconut palm will be unattended when the trees are fallen down. In such cases, after decaying of these coconut bole regions, this will lay the eggs and eggs will be hatched, and then larval period, it will consume that dead and decaying material, crop waste in the field or coconut logs, dead and decaying coconut logs, manure heaps, compost heaps are its breeding sites. There the larva will grow nearly for 6 months. Then the pupation will take place still deeper into the layers of this trunk, rotting trunk, and then adult beetles will be emerged. These adult beetles will be there nearly 16 to 20 days within the pupal shell itself. Later they will come out, and the adult beetle is alive more than 160 days like that. In total, the total life period will occupy so nearly 1 year. For this pest, the package includes number 1, sanitation. That is, the palms which are dead in the garden due to different reasons, maybe lightning, maybe bud rot, or any other cyclones. So those serves as a breeding material. Hence, they are need to be removed from the garden and destroyed to avoid the breeding sites of the rhinoceros beetle. And then if in the young garden, so we can hook out the beetle from the crown region with a small hook, iron hook, and then leaf axil filling with sand and this botanical cakes, pongamia, maybe neem cake, totaling together 250 grams of material. With this, leaf axils are to be filled at quarterly intervals. Sometimes that Clerodendron infortunatum, it is a ornamental plant, shrub. This can be added in compost pits, which will repel the beetles and avoid egg laying in the compost pits. So for this, further step, important biological agent for this rhinoceros beetle is baculovirus. This baculovirus will infect the gut region of the insect, and instead of nucleoplasm, the viral plasm will be multiplied in the gut region, and gradually the insect won't feed on the material and won't transform into the further stages. Likewise, automatically the population will be suppressed. So this baculovirus culture can be mass multiplied in the laboratory. The adult beetles can be inoculated with baculovirus inoculum solution, and those beetles can be released in the garden at the time of dusk at the rate of 10 to 15 beetles per hectare, will naturally suppress the beetle population by visiting of these inoculated beetles with and mixing with the healthy population. The other important bioagent is the fungal bioagent, that is Metarhizium anisopliae. This fungus solution can be sprayed on the compost or manure heaps. With the solution having these spores, 5 into 10 to the power of 11 spores, and 1 liter liquid per 1 cubic meter manure heap will serve as the purpose. And gradually this metarhizium will spread over the other stages of the rhinoceros in the manure heap, and it will result in the total suppression of the life stages of rhinoceros beetle. The other important step is using rhino lure traps. These traps, this lure material is available with many forms, and that has to be arranged in the bucket by using this iron veins like this, and these have to be erected in the gardens. So the attracted beetles can be collected at the same time at weekly intervals or 2 or 3 days interval. These beetles can be destroyed at a time, or again these beetles can also be used for baculovirus inoculation and re-releasing in the gardens. So presently CPCRI is also have developed this rhino lure material, and that PVC pipe lures are available that can also be used in the coconut gardens to suppress this beetle. There are series of experiments conducted in earlier days under NATP project, in which total IPM package was implemented for 1 year, and after 2 years the same garden was monitored for observation of the same location with rhinoceros damage. We can visualize here, this is the garden prior to the implementation of IPM of rhinoceros beetle. So this is the garden after IPM implementation. We can see this all cut leaves in the prior garden. Later it all recovered. These are the new leaves without cuts we can see. So with this IPM package, we can successfully suppress the beetle population in the garden. The other important pest of the coconut is red palm weevil, that is Rhynchophorus ferruginous, and this also more prone to young palms. Sometimes it can also attack adult palms, that is more than 15 years age, and depending on the locality, this weevil attacks. In the early stages, it will attack at bole region. That is this one. In some times, when the palms are grown up, it can attack at collar region or on the trunk region whenever any wounds are there that can lay the eggs with these wounds. So the symptoms, if we observe, the earliest symptom is we can see lower whorls of leaves will turn yellow, and gradually those leaves will be intermittently, few leaves will be getting dried. The other symptoms on the trunk, we can see this pencil size circular holes with brown viscous liquid oozing. Sometimes in serious infestations, chewed fibrous material will also extrudes out from these pencil size holes. We can see the fatal situation of the garden. Almost 90% of the palms are infested with coconut red palm weevil. So we can see the yellowing of leaves, devitalization of the palm, If this situation continues, total tree will be fallen. So this is another further symptom. If we look into the life stages of this weevil, eggs will be laid in the trunk region wherever wounds, soft tissue of the trunk is exposed with the wounds. There the adult weevil will lay the eggs, and larva will emerge from the egg and tunnels inside the soft tissue and feed on the total internal contents of the trunk. And after full grown, it will form into pupa. These pupal cases will be woven with this chewed up fibrous material, and number of these pupal cocoons with chewed up fibrous material we can see in leaf axils and also in the damaged site of the trunk. So from that pupae, adult weevils will be emerged. Total life cycle will take 82 days on an average. So for this also, an excellent IPM was developed for red palm weevil. So regular monitoring of the gardens is much more important for earliest diagnosis of the pest, because it is more serious, and when we neglect, total palm will be going away. That's why regular monitoring is, especially in the young gardens, it is more important. And then crowd planting, more than plant density than recommended spacing, will also result in quick spread of this pest and causing total garden infestation. The other one is periodical and regular crown cleaning will avoid attraction of these weevils. When we see a single palm affected with this or any sort of dead palm in the garden because of lightning, because of bud rot, or maybe any other mechanical damage, this rotting tissue in the stem with this fermented smell will attract lot of number of weevils for egg laying. To avoid that, immediate attention to the dead palms in the garden is more important, and timely treatment. Rhinoceros beetle damage, if it continuously rhinoceros beetle is damaging in the crown, that rotten tissue will attract again this weevil. So timely treatment for rhinoceros beetle is another important management practices, and avoiding injuries on the stem with field tools, agriculture tools is another important precaution we need to take. And then there are different types of traps to attract these weevils in a large number and destruction. That is one natural traps, that is the dead coconut logs can be utilized for this. Small bits, that is 50 centimeters length coconut logs split into 2 halves and smearing with fermented toddy and keeping there as a heap will attract these weevils, and those can be collected and destroyed. Instead of cut coconut logs, we can use coconut petioles also, leaf stalks, that can be made into small pieces, and that can be fermented with the toddy with a small mud pots, and those can also be placed in the garden at the rate of 10 pots per hectare. So next we can collect these weevils and destroy. The other important eco-friendly measure is pheromone traps. Here, small 5 liters plastic bucket wrapped with gunny cloth and making 4 windows below the rim of the bucket on 4 sides will be used as a bucket, and to the lid on the top we can hang this lure material, ferrolure material with required dose which is available in the vials, in the forms as well as our CPCRI institute. And within the bucket, we need to place food bait, which consists of ripened fruit material, that may be pineapple or cocoa flesh or sugarcane, like that, any fermented fruit material, ripened banana at the ratio of 100 grams and 2 grams yeast can be added. And this has to be pre-fermented for 1 day. And next day, small amount of insecticide we can add. And this bucket arrangements we have to make in the garden like this. So periodical inspection of this bucket and collection of this trapped weevils and destruction will help us to eliminate all adult population from the garden. We can see this arrangement to the lid. This is the lure sachet, and this has to be placed on the top, and this arrangement should be made in the garden. And this bucket has to be tied to the trunk of the coconut palm to the more than 1.5 meter or 2 meters height. So in such a way to in the vicinity of the weevils when they visit in the garden. So other thing is, sometimes partially damaged, infested palms will be there in the garden. In that case, we can chisel out that affected portion of the trunk, and that can be destroyed. On the chiseled out surface area, coal tar can be smeared, and sometimes more than half of the trunk would be eaten away. Total hollow trunk will be standing. With the peripheral layer of the trunk, tree will be standing. In such cases, entire internal contents of the trunk we need to be collect, and that has to be destroyed or burnt. And to support the palm without falling down, by adding the cement and sand mixture or some stones, small stones, that concrete material can be filled in the hollowed out trunk. And with that strength, the trunk will be erect and strong, and without falling, it will again can yield fruits. So that is the total IPM of for red palm weevil. The other important enemy to the coconut is Eriophyid mite, that is Escherichia guroronnis. When we look into the history of this Eriophyid mite, the first report it has come from in the year 1960 from Guerrero state of Mexico, and then many Asian and Pacific countries it has caused havoc. In India, it has entered in Kerala first in the year 1998, followed by Tamil Nadu and Karnataka. In Andhra Pradesh, it made its entry in 1999. Since then, so much of work has been carried out in our state. When we look into the that creature body structure, it looks in cream color, and with two pairs of legs in anterior position. And then when you go into details of its size, that is 200 to 250 microns and 40 microns width, and fecundity with 100 to 150, total life cycle takes 7 to 10 days time. The earliest symptom of this mite attack, we can see that white triangular patch on the coconut buttons. And gradually, that white color will turns into yellow-brown and extending its browning on this nut surface gradually as nut grows and as population grows under the perianth. In severe cases, total these fissures will be developed on the nut surface, hardening the coconut husk. In severe cases, gum oozing also we can see, and reduction of nut size. Ultimately, in the earlier stages, button drop, and then not only button drop, this husk will make difficulty in, and copra content will be decreased to an extent of 30%. Not only copra, oil content also will also be affected along with quality. So the preferential stage of the nut, when we examined on different age group of coconuts, the most preferred age is 4 months old nuts, with a population of 502 per 4 mm square area under the perianth. And many studies were made, the impact of, to know the impact of this eriophid mite on different nut characters of the coconut, which are quantity and quality parameters, among which more prominent characters, it will decrease husk weight, which is important in fiber industry, up to 39.2%. And when we see shell, it is also very, very important byproduct and in industrial product, that is 12.3% reduction. In copra weight, 22.9. The other important parameter, oil weight, oil quantity, 30.1% oil quantity will be decreased because of this mite attack. So not only oil quality and content and oil reduction, it will also affect the fatty acid profile of the oils. The impact of weather parameters on the mite population we have studied from taking data of 5 years. It revealed that we are having more impact on the mite population rather than preceding weeks weather parameters. The effect is like this. Temperature is positively significantly correlated with mite population, whereas rainfall and relative humidity negatively correlated. More so with the maximum temperature is more strongly related, and morning relative humidity is more related. So this is highest mite population was also observed during the year, that is when rainfall is less. And when a higher rainfall year, that is in the 2003, a little bit less population of mite was observed. So for this also, the package of management was worked out. Those are, the number one is sanitation, collection and destruction of mite infested and dropped nuts, and then application of organic manures, especially neem cake at the rate of 5 to 10 kg per palm per year, along with other organic manures like green manuring, crops raising and in situ ploughing. So we can see the differences where organic manure is added. Healthy nuts we are seeing, and in the control plot we have less yields. So other important inputs we can make for mite incidence less is regular irrigation along with recommended dose of fertilizer to the coconut palm. to avoid stress to the palm, and raising intercrops, which will create more humid microclimate to which is not suitable for mite population buildup. The other one, as last resort, wherever spraying is possible, we can go for sulfur spraying at the rate of 6 grams per liter of water. And the other thing, root feeding of monocrotophos 10 ml plus 10 ml water will work out. And if we are interested in only botanicals, azadirachtin 10,000 ppm will also work. Or spraying of azadirachtin 10,000 ppm at the rate of 5 ml per liter of water will also check the mite population under the levels. The other natural enemies we have recorded while doing the surveys is Amblyopsis largoensis. These are the predatory mites, and Della distincta. So much of fauna is there, but these are more prevalent. Amblyopsis largoensis and Neocoelus mumai. And entomopathogenic fungi among Hirsutella thomsoni and other Hirsutella species are more prevalent. These can be conjured and make use of for suppressing the mite population. These are the predatory mites, that is, which are active on coconut eriophyte mite. That is one Amblyopsis largoensis and Della species. So the other important pest is coconut slug caterpillar, that is Macroplectra neraria. There are many species in different ecological conditions and different hosts. These three slugs will occur. This is a sporadic pest occurring in localized areas. We can see the severely infested coconut plantations in East Godavari district once upon a time. This is a foliar pest feeding on the entire laminar portion of the leaf, leaving only midribs. Total drying of the leaves and the plants, and sometimes it will attack on nuts also. We can see the slug population on leaflet. It will range. For coconut leaf, there will be, it is a compound leaf in which nearly 180 to 200 leaflets will be there. On single leaflet, it ranges from 6 to 20 caterpillars at a time, sometimes. So we can imagine population per a leaf. Thousands will be there, and total thing will be eaten away within hours. The toxic saliva secreted from the glands of this slug caterpillar will cause irritation to their human skin. That's why it is called coconut slug caterpillar, akutelu in vernacular language. And that toxic saliva will make these spots, dried up patches, leaf spots on the leaf. We can see these coconut leaves. So eating away entire laminar portion, we can see. These are further samples. The other important symptom is this: when this caterpillar stage is there in the foliage on the top, so lot of excretory balls we can see all around the trunk under the canopy. This is another important symptom of this one. And drying because of this attack. Even spindle leaf cannot be spared if the population is high. Gumming on the nut surface, we can see. See, the population of larvae it is spread all over the green. This total life cycle will take 52 days. And eggs will be laid on the leaf laminar portion. Then larvae will come out and feed on the leaf, and pupation will take place in the crown region sometimes, in the leaflet corners sometimes, like this, wherever, sometimes overlapping generations. We can see its fate in the gardens. When it outbreak occurs, work state package of this one, This population not only affects the main crop of coconut, the intercrops, we can see the damage, how they caused. On banana, severe drying. On hedge plants, on intercrops, cocoa, we can see. We can see hedge plants, fence plants, Pithecanthropus species, this one, in the ground level. So after effects, after 1 year, we can see no single green leaf or nut or spathes emerging, total falling of canopy. So in the field visits, we could collect few larval parasites, that is bracken and chrysotid species, and fungal pathogen is very effective, very potential. Up to 90% of the population was suppressed during summer months in the morning hours due to dew forming. It is very congenial for this fungus to attack the slug caterpillar larvae. We can conjure these things to suppress the pest to some extent. The other IPM package thing, sanitation number 1 for any pest, cut and burn the pest affected dried leaves and conservation of these natural enemies. And in severe cases, and wherever possibility is there, we can go for carbaryl. Other thing, this root feeding of monocrotophos will check all the laminar or foliar pests of the coconut. It works wonderfully with that safety rider. This is the sanitation. We can see spraying, which was a community basis arranged at that time during outbreaks in certain areas. We can see immediately after spraying how the population was fallen down. Again, root feeding. Here, clear-cut difference we can see. The farmer who have adopted root feeding, see greenery, saving the foliage, and see other adjacent farmer without root feeding. So total foliage drying, we can see. This is the witness how effective the root feeding. Other important finding from our university with our team we could do is the recognition of positive phototaxis character in the coconut slug caterpillar moths by keeping light traps in the nights in the gardens with 500 watts incandescent bulbs. And other important is checking the population on alternate hosts. These are the studies by this. This is a new innovative at that time in 2009 and 10. We could make that, prove that these adult moths can be attracted to these light traps, and young moths, they can be collected at single light and can be destroyed when the moth stage is prevalent in the gardens, in the outbreak situations. We can see this. So 500 moths in our 1000 moths per single light we have seen here. You can visualize a little clipping, how this was done in the gardens, how the scientists will work for innovative things. All the students can witness. A continuous effort of 1 month during the moth stage in the garden, so will resulted development of this innovation that light traps can be used for controlling the coconut moths. This technology was approved in AICRP itself, and in Kerala state also. In assembly, it was recommended as one of the recommendations for coconut slug caterpillar management in case of outbreaks. See, in entire garden, how the total foliage was burnt up and number of moths per single light. So further refining of this technology is further needed. I would like future budding scientists will work further refining, fine-tuning these technologies. So other leaf-eating caterpillars which were recorded in our ecosystem are leaf-eating caterpillar. This is a Felira species. This is also cause a similar type of damage to the coconut slug caterpillar damage. Here we can see this caterpillar also eats away laminar portion, but only typical difference from coconut slug caterpillar is it attacks top portion of the leaf first. Terminal leaflets will be attacked like this. So entire life stages will come complete within the leaf folds itself. Pupation also takes place in the leaf fold. Nearly 40 days life cycle it occupies. You can see eggs, larvae, pupae, and adult moth. So this is another new leaf-eating caterpillar on coconut. It was identified as Acacia species. This will also cause leaf laminar eating caterpillar. And we can see these eggs, larva, pupa, and adult moth. This will also attack intercrops in the coconut. So this is the life cycle. These are somewhat minor pests, appears occasionally here and there. We can see in the intercrop on banana also. Similar type of caterpillars feeding on the leaf in coconut. So other thing is white grub. That is Leucophilus coniophora. This is more prevalent in Kerala and Karnataka. Not that much, it didn't come across in Andhra Pradesh. But this is important pest in coconut plantations. That's why I'm mentioning. The grubs will eat the rooting system near the bole region and cause damage to the bole region, and the plant will be devitalized and yellowing of leaves and prematuring nut fall, and further spathe emergence and flowering will get delayed, ultimately causing yield losses. For this, repeated ploughing is one of the control measures, and adult beetles can be collected and destroyed, and root zone drenching with chlorpyrifos will effectively works. And another important natural enemy for this is entomopathogenic nematodes, that is Steinernema carpocapsae and other Serratia entomophila. So the other thing which I was mentioning in the starting, coconut leaf beetle, that is the Brantispa langissima. This is not present in India, but waiting around India to enter into India. That's why we need to know this pest. In earlier stages, lot of awareness campaigns were conducted in Andhra Pradesh also. It will cause the damage to the leaves and tender portion of the palm, causing crinkling, this drying and retardation of the palm like this. So these are the typical symptoms. So for this also only natural control, bioagents, conservation and release of those things, and severely damaged palms can be removed from the garden. So other pest, important pest, which has given me chance to visit West Indies, that is Trinidad and Tobago country, that is red palm mite, Ravoella indica. In 1980s, it is there in India, but it was naturally checked with the available natural bioagents in the Kerala state, but it has taken upper hand in other countries. In not only West Indies, in many other coconut growing countries, it was a serious problem. So this is having many names, that is coconut mite and coconut red mite, because of its color. Red date palm, leaf fall spider mite, frond crimson mite, like that, scarlet mite. So it was reported from Caribbean islands in the year 2004 in the first time. So the major distribution of this pest is there in many countries, that is Egypt. Namakawaste, it is there in India, but we are not having it to such serious level. Iran, Israel, Mauritius, likewise, up to Philippines. So many countries likewise. So this, its nature is polypagous. Like our Rugo spiraling whitefly, it is also occurring on many other hosts. That is 32 different palm species it attacks. The major crops, banana, heliconias, ginger, likewise. So its dispersal is through wind, and sometimes coconut souvenirs from countries from coastal regions, and lifting of infested planting material from infested areas to healthy areas, and handicrafts prepared by the leaves, which are infested with red palm mite. We can see souvenirs like hats, baskets, rugs, bowls. Likewise, so many things. Sometimes natural calamities also will bring its invasive species to the other new areas. So when you look into the body structure of this, it is bright red in color and having a flattened spatulate body, and with the presence of this spatulate setae on the dorsal side of the body. And but though it is a mite, there won't be any webbing. But many spider mites, red spider mites, are accompanied with webbing, but thus that is absent in this case. So entire life stages will be in red color only. We can see the symptoms. This RPM colonies will be there underside the leaflet, and those will damage the coconut tissues in the leaf and cause patches or spots, and finally drying. Initially, it starts with localized yellowing, and then total, when all the leaves were infested, total yellowing of the plant. We can see the photographs here, and necrosis symptom, and immature nut fall, total vitality loss of the palm. Sometimes inflorescences may be aborted. Finally, yields will be reduced. So when you look into the life cycle of the red palm mite, we can see the egg, larva, protonymph, deutonymph, and then adult. Total life cycle will occupy on an average 22.78 days by passing through all these life stages within the leaf itself. The other important enemy, all these are invasive pests. That spiraling whitefly, Alirodicus rugio perculatus. It is rampant in Andhra Pradesh now, after entering into Kerala and Tamil Nadu, Karnataka. And this pest will be dealt in detail, my senior colleague, Dr. Chalpatrao, tomorrow, with its biological control. But in the list, enlisting the coconut enemies, you can see the symptoms. It also presents under the leaflet and causing, after sucking, because of sooty mold development, total foliage will appear with the sooty mold darkness. And within the gallery, we can see all stages of this whitefly stages, eggs, pupae, nymphs, and everything. Adults also sometimes we can see. A total package, when we look into the main recommendation from the platform of expert eminent entomologists of India is, number one, not to use chemical control measures with an interest to conserve natural enemies and to save the all fauna which are helpful in suppressing this rugospiraling whitefly population. The things which we can be practice is that preventing of transportation of coconut seedlings from infested areas to the healthy areas, and excellent working the predatory population, that is Dicochrysa aster. Release of these eggs at the rate of 300 to 500 per palm at fortnightly interval is working very good. And then another fungal pathogen, Isaria fimosorosia. During low temperature days, that is less than 35 degrees centigrade, with the colonies of 1 into 108 colonies per unit, that is at the rate of 5 grams per liter of water, along with the sticker material and detergent powder, with the strong jet water spray application, will suppress the life stages of this rugospiraling whitefly. And installation of yellow sticky traps around the trunk with castor oil are working excellently. And for removal of this sooty mold, 1% starch solution spraying. And then other parasitoid, Encartia guadeloupe. This is also working very nicely. And under can't but position, the neem, botanical neem oil can be sprayed at the rate of 5% concentration. And then conservation of these natural enemies is the main step we need to take. And also community-based approach is always excellently suppress this pest. The other minor pest I am going to enlist is this is corid bug, which attacks the tender nuts of the coconut and this type of symptoms on the nut with adults and nymphs sucking from the tissues of the nut. For this, azadirachtin spraying will work. This is not prevalent in our Andhra Pradesh. And the other thing, mealybugs. These will be present under the perianth of the young buttons, and they will cause button drop and drying of inflorescences. Such type of yield losing phenomena we can observe with this. There are 3 species of these mealybugs. We can have this information in script in detail. For the time's sake, I am rushing with the slides for your visualization. The other foliage pest, minor pest is bagworms. Here also different species of bagworms are there. They will eat away all the laminar portion by being in the sachets under the leaf and cause foliage loss. Sometimes in 100 numbers, they will cause severe foliage loss ultimately. Inflorescences loss, button drop, yield losses. The other thing, scales. In young gardens, the scales can attack leaves and cause total yellowing and devitalization and growth retardants. And in young stages, buttons, we can see the layer of these scales. They will suck the sap by being sessile on the surface, causing button drop, yield loss, and growth retardants in the forms like this. The other things, nut borer. It is also not present in Andhra Pradesh, but is present in other states, Cyclodes vomma. And then short hole borer. This is a secondary pest which infests the stem after decayed, after damaged by the Ganoderma wilt disease. This will survive on that. And other inflorescence caterpillars and Musella species is more prevalent in Andhra Pradesh and causing galleries in the inflorescence and causing the inflorescence losses. Other Tarnaka caterpillar is also, once upon a time, it was a great havoc in Tamil Nadu state and causing greater loss to the coconut plantations. The other thing is termites. They can cause damage in the seedling stage as well as in the adult palms and damaging the total stem layers by feeding on the layers of the external layers of the stem. For this, chlorpyrifos drenching around the trunk and rubbing of the galleries with gunny cloth and application of this swabbing with any neem products will work out. So we can see the farmers are not attentive to the termite mounds in the garden. The number one controlling measure in this is they need to dug out termite mounds in the garden and take out the queen like this, and they have to destroy this queen. Otherwise, it will lay thousands of eggs per day, and the next day that soldiers will come out and attack these trunks like this, and total this layer-wise losing of trunk portion, and ultimately weakening of the palm. So other important enemy we are known to us is rat. This will cause in the nursery by eating on the seed nuts in the ground, or in the crown. By being in the crown, they will damage all the tender nuts and cause damage and nut falling like this. For this, crown cleaning is number one management measure. And then using bromadiolone cakes This is an anticoagulant, single dose anticoagulant. These cakes are available in the market as Roban cakes. And these cakes, we can have 3 cakes in the packet. And those 3 cakes can be made into 6 and 2 cakes per palm. In the opposite leaf axils, we can place at periodical intervals. And after observation, after 1 month, if again nut fall is there due to rat damage, we need to give second application of this bromeliad cakes. So clean cultivation will avoid this rodent damage to larger extent. So there are so many other wild boars, monkeys. So scaring away them is the number 1 control measures. And civet bats also there. So all these mammal pests, palm civets. So all these continuous monitoring and scaring away with the mechanical measures will work out to keep away these pests. So nematode is another unseen enemy in coconut gardens. Many times we miss to look into these nematodes. They will cause damage by being inside the roots. And the symptoms which we can recognize this is stunted growth of the seedlings or coconut palm, and premature wilting, yellowing, and purple discoloration sometimes, and curling of leaves. If we look into the roots, so total root system will be weak, and sometimes galls or knots also can be seen on the roots. For this also, excellent biocontrol measures are available. In total, common pest management, what we can observe is this: integration of all possible eco-friendly measures to keep the pests at lower level is the main theme of integrated pest management. The important tools in the integrated pest management is that may be cultural, physical, mechanical, or biological, and botanicals. The final resort is chemical. By integrating all these components, we can make the pests to suppress to greater level and can go with successful crop growing. So in total, always our pest management practices should aim at affecting the life stages of the pest and making the crop to withstand the infestations are main concept for this integrated pest management. That includes, that is in a holistic approach, total integrated nutrient management and pest management practices with, that is using efficient bioagents or conserving the natural enemies, using botanical pesticides, pheromones, and biofertilizers. With this concept of holistic approach, so we can make good production with maintaining or managing agro ecosystems with good biodiversity, biocycles, and soil biological activities. By making all these functions operative, and also in addition to integrated farming systems, that is, we know that multi-tier cropping systems based on the agroecological conditions, different multi-tier cropping systems, if we follow this holistic approach with integrated crop management, integrated pest management, integrated disease management, and integrated nutrient management, will give us very good crop production by avoiding all these pest fauna which are enemies to the coconut plantations. So let us think together for the management with good thought and share the information among the farming community or scientific community on agriculture or horticulture sector. With this background knowledge, built up knowledge, we can sustain our Kalpa Vriksha, which is the main source for our livelihood of mankind. So we can throw some light in future on which we can concentrate for successful coconut crop cultivation. That is identification and conservation of local and potent strains of natural enemies for specific pests, or in common in total pest fauna of these coconut plantations, and those collection, isolation and mass multiplication and field evaluation of these microbial or biological agents, whatever it may be, against coconut pests, and identification of effective botanicals, and studying in depth on the influence of crop habitat or ecology and its diversity on the pests and natural enemies, and including this development of synthetic pheromones and identification of resistant gene against coconut pests will make us successful farming things, and we can have such a type of good yields from our Kalpa Vriksha. I am very much thankful for giving me this platform for the organizing committee and also faculty, and with the patience hearing of all the student community. Thank you very much.
Thank you, madam. So participants, if you have any questions, you can unmute yourself and you can ask now. We didn't receive any questions in the chat box also, madam.
Okay.
Live also you check once.
I have checked
Pardon?
live.
Live?
In live also there are no questions, madam.
Okay.
I think they got a complete knowledge about coconut pest. That's why
They don't have got any idea.
No, madam, not like. They understood every point which you have explained. That's why they didn't ask anything. So, on behalf of organizing committee and all the participants, I express our sincere thanks to you, madam, for enriching the participants with different coconut pests and their management. So, your experience and expertise in coconut crop gave an insight into different pests of coconut, such as coconut black-headed caterpillar, rhinoceros beetle, red palm weevil, aphid mites, caterpillar, etc. Their symptoms of damage and also management of all the pests in coconuts. Once again, thank you, madam, for sparing your precious time for all of us. Thank you, madam.
Okay, thank you very much, and you are most welcome.
Thank you. Shall I close this session, madam? Okay.
Thank you, Amma. Kalpana, Padma, Naram Naidu, Sujatha, Uma Jyoti. Youngsters, I don't have familiarized with names.
Thank you, madam. Thank you. Very nice lecture, madam.
కొత్త వాళ్ళని
Ravikiran and Ranjit. Bhaskar.
వీడియోలు తెరిస్తే అందరిని ఒకసారి చూస్తా, ఫోటోలు కాకుండా అసలు బొమ్మలు పెట్టమని. స్ట్రీమింగ్ ఆగిపోయింది కదా.